A very good morning to one and all. The topic for my poster is a review on salivary metabolites and mass spectrometry in oral squamous cell carcinoma. Oral squamous cell carcinoma, it is a malignancy of epithelial origin. It refers to carcinoma occurring inside the oral cavity. Etiology is multifactorial, and oral cancer is still the sixth most common cause of death. And according to Globocon, in the year 2020, India recorded the total number of cases as 1,35,000, with males alone constituting 1,04,000. The numbers are constantly increasing and the survival rate is very less in spite of treatment. That is because of late detection. We have different methods of investigations. We have staining techniques, exfoliative cytology techniques, incisional biopsy, and also imaging techniques. But they all are associated with their own shortcomings. All the techniques, they involve very skilled professional, and there should be a proper area of identification when we take the samples. An adequate amount of sample should be taken, and especially when it comes to incisional biopsy, the procedure, since it is being invasive, it requires a lot of convincing of the patients. We have different imaging techniques ranging from CT scan, MRI, PET, and ultrasound, which are being profoundly used. But you find the sensitivity of these techniques, they range from 52 to 66%. It is because of this reason we find the oral squamous cell carcinoma patients are being either undertreated or they are being overtreated. Different omics technology are used in analytical field to study about cancers. The latest in omics is metabolomics. In metabolomics, we study about the basic metabolites, sugars, nucleotides, amino acids, and lipids. The advantage of metabolomics when compared to other omics is, when all other omics tells us what may happen with the cell, metabolomics tells us, or the metabolites tells us, what is the current status of the cell. And when we study about a particular gene or a particular protein, we should know that one particular gene that we study is actually involved in multiple other reactions simultaneously. And what we study with the proteins, those proteins are actually subjected to post-translational changes. It is because of this, we will not be able to know the exact impact of the gene or the protein that we study on the phenotype of the cell. But the advantage of the metabolomics or the metabolite is that we study about the end product of all the reactions that are happening in the cell. So irrespective of all the changes that is happening in the upper stream, the end products, they are still the same, and metabolomics study is only that. Another advantage associated with the metabolome or the metabolomics is it studies the molecules that are exactly at the interface between the human body, the internal structure, and also the environmental influences. And anytime in oral squamous cell carcinoma, we know in pathogenesis, it is not only the internal makeup, but it is also the external environmental factors that influence a lot in the pathogenesis of oral cancer. The science of metabolomics is studied by using two analytical tools like NMR and mass spectrometry. NMR is nuclear magnetic resonance, and mass spectrometry, it can be either gas chromatography or it can be liquid chromatography mass spectrometry. To mention few words about the difference in these two NMR and mass spectrometry, they are. NMR brings about good details about the metabolites. It does not involve greater sample preparation, and it can identify the molecules from the concentration of micromolar to millimolar. And we find the sample that are analyzed, they are not destructed in the process of analysis. But this, it is associated with very less sensitivity because of peak overlap. But when it comes to mass spectrometry, though the sample preparation is going to be very high, involving procedures like extraction, derivatization, especially as in case of gas chromatography mass spectrometry, we find the detection limits of the molecules, the range from the concentration of picomolar to micromolar, and also the sensitivity is very high and high number of molecules are being detected by them. To mention few words about the mass spectrometer, any mass spectrometer consists of three components: ion source, that is used to ionize the molecules; mass analyzer, that is used to segregate the molecules based upon the mass over charge ratio; and detector, that is used to produce the measurable signal. This poster focuses on the trio, metabolomics, saliva, and oral squamous cell carcinoma. Now coming to the saliva part of it, saliva is considered now as a very safe sample that can be used in the biological analytical process. It is obtained by non-invasive method. It is rich in biological information, and this requires very minimal training for the volunteers for them to collect the sample, so it can be used even in mass camps. So the sample that is collected, saliva that is collected, it can be either the whole mouth saliva or it can be a specific gland saliva. When we say whole mouth saliva, it refers to that component, that saliva which has got the glandular fluid obtained from the glands. It also includes the gingival crevicular fluid, and it also involves cellular components, which mainly consist of the shed epithelial cells, the oral bacteria, and also the immune cells. Specific gland saliva, it refers to the saliva that is collected from specific gland, as it is obtained from as in case of parotid gland by Lashley's cup, etc. And the type of collection also depends. It has got different methods. It can be obtained either by chewing method or taste stimulation, or it can be obtained by spitting, or it can be obtained by drooling method. Salivary metabolomics is used in the study of oral squamous cell carcinoma. Mariana et al. Used GCMS to identify the metabolites to differentiate between normal and oral squamous cell carcinoma. Malic acid and maltose and other molecules were identified to differentiate between the groups. Similarly, Sridharan et al. Used LCMS in Indian population to differentiate between normal and potentially malignant cases like oral leukoplakia and also oral squamous cell carcinoma. They identified molecules like 1-methylhistidine can be used to differentiate. Study by Ishikawa et al. And Sujimoto et al. Also identified molecules like indole 3 acetate and ethanolamine phosphate can be used to differentiate lichen planus and oral squamous cell carcinoma from normal patients. In spite of all the research work that are being carried out, we still have challenges that are to be met. As in case of saliva, the time of collection, the type of collection, and oral microbes, they all play a very important role. Regarding the time of collection, it is important because 15% of the metabolites that are seen in saliva, they are supposed to exhibit a circadian rhythm. So we should be very particular about the time of collection. The type of the collection is also very important because it is seen saliva that is obtained by stimulation using citric acid. They have shown decrease in the majority of the metabolites. And oral microbes constituent should also be studied prior before collecting samples because we should be they also secrete metabolites, and we should be able to identify the metabolites secreted by them and those secreted by our own body cells. And in general, when it comes to metabolite, we should be able to understand, or we should have a clear idea that they have a tendency to vary depending upon the geographical area. It tends to vary with ethnicity, their lifestyle, especially following exercise or any habits like smoking, alcoholism, or even following medication. And when it comes to analysis, again, right from the start, like how the saliva is being stored, the temperature at which it is being stored, the type of the solvents that I use, the mass spectrometer that is used, and the statistical method, the type of the metabolite or the concentration of the metabolite can vary. So taking all this into consideration, we should be able to design a proper methodology and then carry out the research. So I would like to conclude the poster mentioning the need is we need to conduct large multi-centric study and standardization of the protocol should be established. So when this is done, a proper sensitive diagnostic method along with appropriate treatment which complements that can greatly improve the survival rate of the oral squamous cell carcinoma patients by almost 90%. I would like to thank the organization for giving me an opportunity to present the poster here. Thank you all.